Okay, good afternoon again, everyone. So, we are the reporters for the cell division and checkpoints. I am again Kaiel, Millicent, Quintero, and together with Ma'am Rose Ann Pa-alisbo, we will be tackling the different topics included. In the first one is cell division, which is including the mitosis and meiosis, and of course, topics included in the checkpoints, which are found, no, in the different phases in the cell cycle. So let us start. First of all, these are the list of the topics that we will be discussing. So the first one is the introduction to the topics. The second one will be about cell cycle and what are the different phases that are included in the cell cycle. The third one will be the checkpoints. Now, the checkpoints are the regulatory processes in the cell cycle, and they are also found within or in between the phases of the cell cycle. And for the fourth one, we will be tackling about what happens in the mitosis process as well as in the meiosis process. And for the application, we will be differentiating what is spermatogenesis from oogenesis, and that will be followed by the conclusion of the report as well as the summary of the whole report. Okay? So I will be the one discussing the introduction, the cell cycle, the different checkpoints, and the mitosis, and then Ma'am Pa-alisbo will be the one discussing meiosis, differentiation between spermatogenesis and oogenesis, as well as the conclusion and the summary. Okay? So let's start with the introduction. Okay, every living thing contains genetic material. Okay, and genetic material, when we say genetic material, the first word that will come to our minds is either the DNA or the genes. Okay, so our DNA is actually composed of linear segments of genes, and the genes are found inside the chromosome. Okay, now the chromosomes are actually playing a very important role in the cell cycle or cell division, because your chromosomes, they serve as vehicles for transmitting genetic information. Now, transmitting genetic information in the sense that this includes the distribution of chromosomes and also the replication. So replication of chromosomes and as well as its distribution is the main point of the mitosis and meiosis, or the whole cell division process itself. Okay? So the manner in which chromosomes are transmitted from one generation of cells to the next and from organisms to their descendants must be exceedingly precise. When it comes to cell division, there is no room for errors and mistakes, because such errors and mistakes might result to further mutations as well as formation of tumors. So we don't want that. And it is really amazing how this type of process, the cell division, is very precise in doing their job. Okay, next. Okay, genetic continuity between the generations of cells and between generations of sexually reproducing organisms is maintained through the processes called mitosis and meiosis. So, mitosis and meiosis, their role is to maintain, the genetic continuity. Okay, so in this discussion, we will have a glimpse on, number one, how genetics is involved in cell division, or maybe specifically how genetics control, the cell cycle or cell division. And number two, how cell cycle continues through passing different checkpoints. So what are the roles of these checkpoints in maintaining a precise replication of DNA and the distribution of chromosomes from one generation to another? Okay, so let's first discuss the cell cycle. When we say cell cycle, this involves many cells that are undergoing a continuous alternation between division and non-division phase. So when we say cell cycle, these are the events that occur from the completion of one division until the completion of the next division. So this constitutes a process called cell cycle. Now, the cell cycle is actually composed, generally composed of the interphase and the mitosis or mitotic phase. And the interphase is furthermore divided into three phases. Okay, namely the G1 phase or the gap 1 phase, followed by the synthetic phase, okay, or synthesis phase, and followed by your gap 2 phase or G2 phase. After the interphase, mitosis is initiated. So mitosis will follow the interphase, and your mitosis is composed of four main phases also, namely your prophase, your metaphase, your anaphase, and telophase. Now, if you will count them, a total of four phases are included in the cell cycle: the G1, the S, the G2, and the M for mitosis. But the G0 stage is not included here, no? So we can say we have a total of 5 phases. But G0 stage is not included because, again, not all types of cells will enter the G stage. This is only for the cells that enter the G0 stage is only for damaged cells and also for very specific types of cells. So, hindi lahat nakakapasok sa G0 stage. So, okay, but we will still include the discussion for the G0 stage. Okay? So, let's first discuss the interphase. Now, in the interphase, there is an absence of visible chromosomes, no? Because only chromatin fibers are included. Now, in the interphase, biochemical activity, there is a involvement of biochemical activities that are devoted to cell growth and for normal function. So, your interphase also involves the replication of the DNA of each of the chromosomes, which is very critical, no? It is a very critical step for initiating mitosis. Okay? So, your interphase is composed of G1 phase, S phase, and G2 phase. So, as you can see here, this is the G1 phase. This is the S phase and the G2 phase. Among the three phases, S phase is the most time-consuming. Okay? So, in the G1 phase, this is generally for cell growth. Okay? So, there is no DNA synthesis or replication in the G1 phase. So, walang DNA replication in the G1 phase. Now, the G1 phase, usually it takes 5 to 8 hours in order for this to finish. So what happens in the G1 phase? The first one, in the G1 phase, there is production of organelles. Okay? The production of the organelles must be enough for 2 cells because remember, in the end of the mitosis, we will be producing 2 daughter cells. So there must be enough organelles for 2 daughter cells, and that those organelles are produced in the G1 phase. The second process that is happening within the G1 phase is the synthesis of proteins and enzymes. So these proteins and enzymes are used later on in the replication of the DNA in the S phase. Okay? So that is the G1 phase. Now, as you can see here, I included 2 types of pictures because in this one, G0 stage is not included. So, dalawa yung nilagay ko, no, na picture. So as you can see here, before proceeding with the G1 phase, you will see the G0 stage. Now, the G0 stage, cells that are entering the G1 phase will actually choose between two paths. Okay? So the first path is that they will uh, continue with the cell cycle. They will proceed to the S phase. But the second path that the cells will be choosing, no, if they will choose, is entering the G0 stage. Now, when the cells enter the G0 stage, they will become quiescent, or in other term, dormant. Okay? So the cells become dormant, but they are still viable. When we say viable, they are still metabolically active. Okay? So there is metabolic activity in the cells that are entering G0 stage, but the difference is that they do not proliferate. Okay? So when a cell enters the G0 state, it will stop dividing. Okay? So, we have an example which we call the cancer cells. So the cancer cells actually, they are very proliferative. So they will avoid, no, as much as possible, they will avoid entering the G0 stage kasi nga pagdating ng G0 stage, walang proliferation na nangyayari, walang cell division. So either they will pass the G0 stage very quickly, or they will not enter G0 stage at all. Okay? And then some cells naman, they will enter G0 stage and they will never leave. Okay? But that is for only specific types of cells. Okay? So let's go to the next phase, which is the S phase. As you can see here, the name is DNA replication. Okay? So the S phase or synthetic phase is very important in preparation for the entering to mitosis phase. Why? Because in the S phase, coming from its name, synthesis, this is where your DNA is being synthesized. Okay? So this occurs before the cell enters mitosis. Therefore, if our parent cell is diploid, meaning 2N ang kanyang DNA, for example, 46, pagdating ng S phase, nagiging 4N yung kanyang DNA amount. Okay? So, nadodoble yung ating diploid DNA. So, ang mangyayari from 46, after niya dumaan ng DNA replication in the synthetic phase, nagiging 92 chromosomes yung present. Okay? So that is the purpose of the S phase. We are synthesizing number of chromosomes that will be enough for 2 diploid daughter cells. Okay, so from 2N, nagiging 4N, yung dito sa synthetic phase. Now, after the synthetic phase will be the G2. Okay, G2 or GAP2 phase. In GAP2 phase, there's also no DNA synthesis. Okay, the purpose of the G2 phase is for preparation for mitosis and also for growth. When we say growth, cell growth. So the cell must increase in cytoplasm. Because remember, we will be producing 2 daughter cells. So the cytoplasm must be enough, no, for the dividing into 2 daughter cells. Okay, so we will be increasing the amount of the cytoplasm, and then the volume of the cell must be doubled in the G2 phase. And in the G2 phase, the initiation of mitosis will start. After that, we will be going to the mitotic phase itself. Now, in the mitotic phase, there is a division of a parent cell into 2 daughter cells. Now, what is the main goal of mitosis? Mitosis for example, in a zygote, no, it is the foundation for development and growth of organisms, especially the zygote. on the other hand, in the adult organisms, the use of the mitotic phase is basically for wound healing and also for forming or replacing cells in specific tissues that are damaged. So, yun ang kanyang function in the adult organisms. Okay? So, these are the hours that the different phases in the cell cycle will take. So, G1, maybe 5 hours. Sa S phase, it will take 7 hours. In the G2 phase, siya ang pinakamaikli, 3 hours only. And then in the mitosis, less than or equal to 1 hour. Okay. So, in the prophase, 36 minutes, siya ang pinakamahaba. In prophase, yun pala. In metaphase, 3 minutes only. In anaphase, 3 minutes only. And in telophase, 18 minutes. Okay, so that is the cell cycle. Now, these are what is happening in the G1 phase, the synthetic phase, and the G2. Okay. Okay, next. Let's go to the cell cycle checkpoints. Now, the discussion of the cell cycle checkpoints very important, especially when we are talking about genetics and the regulation of the cell cycle. You can also call your cell cycle checkpoints as cell cycle regulation. Okay, we are regulating the flow of the cell cycle with the use of different genes. Now, cell cycle is actually governed by a genetically regulated program that has been conserved throughout evolution. Disruption of the regulation of your cell cycle may underlie the uncontrolled cell division characterizing malignancy or leading to formation of tumors. So, kapag nasisira yung cell cycle regulation or nawawala yung mga checkpoints, okay, in the cell cycle, it will lead to production of tumors because we cannot control anymore the they call this the proliferation of cells. Okay? So, we have 3 checkpoints in our cell cycle. The first one is the G1 to synthetic transition. The second one is the G2 to mitotic phase transition. And the third one is the axis or mitotic phase transition. Okay? Now, okay. So, I will discuss another, Ito, siguro. When the proteins that regulate the cell cycle are mutated or absent, cell can divide uncontrollably, leading to cancer. When there's production of tumors, expect na natin na merong cancer. Okay? So, we actually have 2 different types of regulation when it comes to checkpoints. So, the first one is regulation by external events. When we say external events, this includes the hormones. So, when we say hormones, example of that is the human growth hormone. When there is lack of your HGH, ang nagiging result niya is dwarfism. And then, kapag meron namang too much hormone of HGH, ang nangyayari is gigantism. So, that is one example of external event. Another one is crowding of cells. When there is too much, or when there is crowding of cells, it can inhibit cell division. And the third one is the size of the cell. When your cell grows, nagiging inefficient siya due to its size. So, the solution for that is through cell division. Okay, so those are external events that regulate your cell cycle. How about the internal checkpoints? So, internal checkpoints, ang nangyayari dyan, when we produce daughter cells, it must be, or the duplication of the DNA must must be exact with the parent cell. Now, if mistakes in the duplication or distribution of chromosomes is happening, there will be mutations that may be passed forward to every new cell produced from the abnormal cell. So, the function of your checkpoints is actually to prevent the proliferation of compromised cells. Ipe-prevent ng checkpoints yung pagdami or yung pag-divide ng mga cells that contains mutations. So, kaya nga siya checkpoint. It is checking, para siyang checkpoint sa road. It is checking whether a cell has compromised chromosomes or duplicated chromosomes. How do checkpoints do that? The checkpoints will actually hold the progression of the cell to the different stages of your cell cycle until conditions become favorable. Okay? So, let us discuss the different checkpoints in the cell cycle. We have the first one, G1s Checkpoint. So, ito siya. The cell growth checkpoint. Your G1s checkpoint occurs towards the end of the gap 1 phase. Yan. So, until sa simula ng synthesis. So, this actually, the function of the G1s checkpoint is to determine whether all conditions are favorable for cell division to proceed. So, your G1s checkpoint checks for presence of genomic DNA damage. Okay, it is checking whether the cell is big enough and has made the proper proteins for synthesis phase. So, what will happen if there is actually a mistake, then what happens is the cell will go through a resting period or it will enter the G0 stage until a favorable condition will occur. So, saka pa lang siya. Or kapag naayos na yung mga mistakes, so saka pa lang siya makaka-proceed to the synthesis phase. Okay? Now, the second checkpoint is the G2m Checkpoint. This occurs during the synthesis phase itself. So, ito siya, nasa synthesis. Now, your G2m checkpoint actually bars, or blocks the entry to mitotic phase, okay, if certain conditions are not met. It actually ensures that all of the chromosomes have been replicated and the replicated DNA is not damaged. Okay? So, if problems are detected dito sa ating G2m checkpoint, what will happen is the cell cycle is halted. So, matitigil yung cell cycle and the cell attempts to either complete the DNA if kulang yung DNA na na-produce or repair the damaged DNA. Okay, if there's a problem in the duplication, it will try to repair. Now, if naayos, if everything is naayos na, the cell will continue to the mitotic phase. Okay? Next, we also have a checkpoint found in the Metaphase. Okay? So, sa mitotic, you call this as Mitosis checkpoint, which is found in the Metaphase. Okay, so this occurs during the mitotic phase in the metaphase. Okay, now you can also call this M phase checkpoint as spindle checkpoint. Bakit spindle checkpoint? Because it actually determines whether all the sister chromatids are correctly attached to the spindle microtubules. So, diba, the microtubules are the ones that attaches to the sister chromatids in order to pull the sister chromatids to the center and also to pull the strands of the chromatids to the opposite sides. Now, the separation or the disjunction, we call that actually as disjunction, the separation of your sister chromatids. This happens in the anaphase, and it is actually an irreversible step. Hindi na, when the sister chromatids separate into the different poles, okay, this is an irreversible step. So, the cycle will not proceed. Yung cycle natin dito, from the metaphase, it will not proceed to the anaphase step until the kinetochore of the sister chromatid is not firmly anchored to at least 2 spindle fibers. Okay? So, that is the third checkpoint. So, we're done with the checkpoints. Now, let's go to the differentiation between mitosis and meiosis. Okay? So, the first process involved in genetic continuity, okay, or passing of genetic information from one generation to another, is called the mitosis. Your mitosis provides mechanism by which chromosomes, having been duplicated, are distributed into progeny cells during cell reproduction. When we say progeny, the following cells. Okay, now, your mitosis converts a diploid cell into 2 diploid daughter cells. Galing sa isang diploid parent cell, you can produce 2 diploid daughter cells. And each of the daughter cells are having the same number of chromosomes as the parent cell. Okay? So, the daughter cell is like a twin, no? From a twin of the parent cell. They do not differ in size, they do not differ in cytoplasm amount, and they do not differ especially in the number of chromosomes. Okay, if there is a difference in the number of chromosomes between your parent cell and daughter cell, if naiba yung number of chromosomes sa daughter cells, then maybe there is a problem in the replication of the chromosomes or in the distribution of the chromosomes. Okay. You can call meron tayong abbreviation for the phases inside your mitosis. Na-include na dito yung interphase. So, IPMATC. IPMATC, that stands for Interphase, Prophase, Metaphase, Anaphase, Telophase, and lastly, the cytokinesis, or the division of your cytoplasm. Okay? IPMATC. So, let's take them one by one. So, here is a picture showing the whole process involved in mitosis. Okay? So, ayan siya. The first one is the G2 of interphase. Now, in the interphase, the chromosomes are extended and uncoiled. Diba? So, wala pang prominent chromosomes in the interphase. What you can see is actually the chromatin. They are extended and uncoiled. So, this is the chromatin. So, this chromatin here is actually duplicated. Hindi pa prominent yung mga chromosomes, pero actually, the chromatin here contains genetic material or number of chromosomes enough for 2 daughter cells. So, in short, we have ang diploid number natin dito is 4n. I mean, the number or the amount of chromosomes here is not diploid but instead 4n. Kasi kapag diploid, diba, 2n. Dito, 4n tayo, meaning it contains 92, total of 92 chromosomes. Next, in the what follows your interphase is the prophase. In the prophase, chromosomes coil up and condense. Nagiging mas condense yung ating chromosomes, and also the centrioles will start to move apart. Okay? They will start to move into the different poles. So, over half of the mitosis is spent in your prophase. Ito siya ang pinakamahaba, among the phases. Now, in the prophase, your nuclear envelope and nucleolus begins to break down. Nawawala dahan-dahan yung nucleolus and nuclear envelope. And they gradually disappear. Okay? So, next, we will go to the pro-metaphase. Okay, balik tayo sa prophase. In the prophase, we still have 4n as a number of chromosomes, so 92 pa rin. Pagdating ng prophase, pro-metaphase, okay, the centromeres align on the metaphase plate. Or no, the chromosomes are clearly double structures na. Sorry for that. In the pro-metaphase, we can see clearly the structure of the sister chromatids. Okay, so these are the sister chromatids joined together by the, what do you call this? By the centromere, and on the centromere we will see the kinetochore in which the spindle fibers will attach. Okay, so in the pro-metaphase, very clear na or prominent na yung ating double structures of the chromosomes or sister chromatids. And the centrioles had already reached the opposite poles. So, nakapwesto na yung centrioles natin on the opposite poles. And then, slowly but surely, the spindle fibers are finally formed. Okay? These are the spindle fibers. Next, on the metaphase, ito na siya. On the metaphase, the centromeres found on the sister chromatids will align on the metaphase plate. So, yung sister chromatids natin will slowly align at the center of the metaphase plate. You call this one a line that is perpendicular to the axis is the metaphase plate. Okay? So, magkaroon na ng alignment sa metaphase, and then the kinetochore, okay, you can see here the kinetochore, kanina nabanggit ko. The kinetochore actually plays a very important role in preventing errors in chromosome migration or distribution. Because your kinetochore again is the structure in which your spindle fibers will attach. Now, kapag naka-attach na yung kinetochore doon, or naka-attach na yung spindle fiber sa kinetochore, ang nangyayari, there is pulling of the sister chromatids apart. Nagsiseparate yung sister chromatids. Now, when the kinetochore is altered, it will cause error in the separation of the sister chromatids, and it will lead to the alteration of the diploid content of the daughter cells. Kapag nagkaroon ng error sa separation ng sister chromatids, we might not produce a diploid daughter cell. So, the presence of the kinetochore and the attachment of the spindle fiber to the kinetochore is very important in preventing mutations. Okay? Next, let's go to the anaphase. Now, in the anaphase, the centromeres actually split apart and the daughter chromosomes migrate, you call this structure daughter chromosomes. Kanina, sister chromatids, this one. Kapag naghiwalay ang sister chromatids, it will form two structures called daughter chromosomes. Okay? The daughter chromosomes will migrate to the opposite poles. And this is a very critical stage for chromosome distribution. Ang process ng paghiwalay or separation ng sister chromatids natin is called disjunction. Okay? Now, there's a difference in the number of chromosomes here. Kasi diba, naghiwalay na yung ating sister chromatids. So, therefore, meron na tayong dalawang set ng diploid chromosome. Okay? So, ang mangyayari sa isang cell, may dalawa ka ng set ng 46 chromosomes. 46 on the first pole and 46 on the second pole. Okay? The last the last phase in the mitosis is your telophase. Okay, in the telophase, daughter chromosomes arrive at the poles. Okay, and okay, no. First, there is a reversal of events that occur during prophase. So, in the telophase, i-reverse natin yung mga processes like condensing of the chromosome. Ang mangyayari dyan, the chromosomes begin to uncoil and become diffuse chromatin once again. In the telophase, the chromosomes will become chromatin again, and the nuclear envelope and nucleolus again reforms. Okay? So there is appearance again of the nuclear envelope and the nucleolus. And after that, the telophase will initiate the cytokinesis, or the division or parting of your cytoplasm. Okay? So, ang nangyayari, after the cytokinesis, after the mitosis, the mitosis already formed two diploid daughter cells. Okay? So, let's see here if we can identify the different phases. So, saan dito ang interphase? Okay, can you identify the interphase here? So, the interphase here is this one. Ito siya. Okay? So, how many chromosomes are present in the interphase? Let's try to... No? Okay. So, in the interphase, we have 4N. This means, ang number ng chromosomes natin is actually 92. Okay? Next, where is the prophase? So, the prophase is this one. Okay? Ang prophase natin is this one. There is condensing of your chromosomes. So, ang total ng chromosomes in our prophase is also 92 chromosomes, 4N. Next, where is the prometaphase? The prometaphase is this one. Okay, there is formation of spindle fibers. So, the number of chromosomes is also still 92 chromosomes, or 4N. Where is the metaphase itself? Malalaman mo ang metaphase kasi lahat ng sister chromatids is found on the metaphase plate. So, ito siya, metaphase. And the number of chromosomes is still 4N, or 92 chromosomes. Followed by your anaphase. So, pagdating ng anaphase, there is separation of the sister chromatids. So, therefore, ito siya. Yan. So, your anaphase, medyo magkaiba na ang definition ng kanyang number of chromosomes. 2N on each pole. Therefore, there is 2 sets of 46 chromosomes on each of the pole. Okay? And then lastly, we have the telophase and cytokinesis. So, in cytokinesis, you can see here formation of the separation of the cytoplasm. So, telophase is not difficult to find or locate. So, in telophase, you are producing 2 daughter cells, each with diploid number of chromosomes. 2N, meaning 46 for this one and 46 for this one. So, that is the final product of your mitosis. So, this is the end of my report. The meiosis part will be reported by Ma'am Rose Ann Pa-alisbo. Thank you very much for listening, and I hope that somehow may nakuha tayong information from my report. Okay? So, thank you very much, and see you next time po. God bless and goodbye.